Thanks. So I was telling y'all that when I first started my job, Michigan, I actually moved in January, and it's November right now, but it feels like January. But what I will tell you is it's 24 degrees outside, but the thing I remember the most about being in Michigan was like the warmth of the colleagues. And I felt like you guys have been super warm with me today, so I think your warm makes up for what's outside. So thank you. So thank students at Valley for talking with me. And I'm not sure you caught the little pun in Al's introduction. She meant reproduce, and I'll be talking about reproducibility today. Yeah, just a little bit of back. So I always, want to get the collaborators out first because it's really important. I think you can acknowledge them at the beginning. So this is joint work, and as Al mentioned, I've been since here I've moved to Penn State and now at University of Colorado. So in fact, this is actually work that started back when I was at Penn State. So Daisy Philtron was a PhD student who worked with me, and the methodology I'll be talking to you mostly about is basically from our dissertation, and that was joint work with Yafei who's a bioinformatics student at Penn State, and Qunhua who was my colleague in statistics there. And then I will have a little bit of time to talk a little bit about metabolomics, which is how Al and I have connected, and that part's going to be joint with several people in my department, including Debashis Ghosh, Weiming, Zhang, and Katerina Kechris. Okay, so you've probably heard about this reproducibility crisis in biomedical research. We've seen studies retracted, withdrawn because they weren't reproducible, and you sort of see that sort of happening at a higher rate. So I think it's fair to say that we need better methods to enhance and evaluate reproducibility. But it sort of begs the question: is what does reproducibility mean? Because it's one of those things where that word means different things to different people. So I thought before I talk about the way I think about reproducibility for today's talk, I'd give you some other examples of reproducibility. So sorry for you can't see the screen here, but this is actually reproducible epidemiologic research from a paper by Roger Penn. So he's a professor at Johns Hopkins, who's written quite about reproducibility, and he had sort of a four-step process for reproducibility. Again, probably hard to read from where you're seeing, but basically the idea would basically you have data, methods, documentation, and distribution. So he goes for the most extreme case, where basically for him reproducible data basically means you actually, as a reader, get the same data set that the authors analyzed. Okay. Methods, obviously, there's okay, so with methods, there's the analysis you run, and potentially you should be able to reproduce that as well. And then not only is he's arguing for not just the methods, but also having documentation and distribution. So documentation would say something about like why you made the certain choices and model assumptions and what models you fit and why you fit them. And then distribution is to sort of let people access the software. There's a second framework, and this is one by Victoria Stodden, who's at the University of Illinois. And she describes various kinds of reproducibility requirements. So the first one basically is reviewable research, and that's basically saying, that's the one I think we most of us engage in. When you read a paper, hopefully the authors have written a method section where they describe how they analyze the data. You look through that method section, and you basically try to see if the results at the if they look credible. So that'd be reviewable research. And again, as students, as faculty, we review stuff, we review papers, review grants. So that's one we engage in a lot. Point two is probably more what the way Roger was defining on the previous slide, which is replicable research. And basically this would be that you have all the materials and tools made that would allow one to duplicate the results of the research. And again, how many of you are familiar with R Markdown? A few hands. How about Jupyter Notebook? More hands. Those are examples of tools, right, that potentially allow you to do replicable research. Confirmable research, you would basically, the main conclusions would could be attained independently without the use of software provided by the author. So confirmable is kind of what a lot of us do anyway, right? You get a finding, let's say you're a basic scientist, you get a finding from a paper, you might have the same organism or system in your lab, and you basically see, can I do the experiment the way described in the paper and get the same thing? That'd be sort of the basic science version of what it means to be confirmable research. Auditable research would basically audit means like you leave a trail, like a paper trail, and this would basically be you have data and software archived so that research can be defended later. So how many of you are familiar with something called GitHub? Sounds good. So GitHub is basically something that has something called version control in it, and the version control basically allows you to do exactly this auditable research path. And then the last one would be open and reproducible, where you basically make it open to everybody. So those are two notions of reproducibility. We're not going to actually cover either of those really today. We're going to do something much more basic. And the setup that we're going to be looking at is you have a high dimensional omics data set where you measure things, where you have the same sample measured 2 times. So that's like the most basic problem in a very basic problem. So this is not a big data set the way Ala was describing. This is like 2 samples. So obviously one way you could sort of assess the reproducibility is to look at the correlation, right, between the 2 samples. And again, there's sort of a finding by my colleague Qunhua, where basically some genes would show high correlation and other genes would have almost no correlation. So she had developed something called the irreproducible discovery rate, or IDR. And if you're familiar with, again, one common quantity we see quite a bit in bioinformatics is false discovery rate. So how many of you ever used like a Q value for doing stuff? I see hands, good. How about Benjamin-Hilbert procedure? Does that ring a bell? Some other hands? So yeah, so those are examples of methods and procedures that control the false discovery rate. And this was sort of extending that idea to looking at irreproducible false discovery rate. And basically, there's a modeling approach done by Qunhua, and that was based on a mixture of copulas to estimate the IDR. And that's kind of our kind of setup that we'll talk about later. So here's an example from a picture from her paper about how this worked. And basically, IDR. So it's basically of the discoveries you find, what fraction of them do you expect to be irreproducible? And irreproducible obviously is a bad thing, right? That means unable to reproduce. So this is actually from a chip or chip-seq. Yeah, no, it must be a chip. A chip experiment. I'm not sure if it's seeker or a chip, but basically this is looking at different peak colors for finding finding peaks in the chip data. And basically what you see is, some algorithms, on the x-axis, you basically have the number of significant peaks, and then the y of the IDR. So again, with IDR, the larger the number is, the worse that is, right? So you basically see, I mean, by definition, the more things you're calling, on average, your IDR is getting worse. So what really matters is like how fast do you rise. The algorithms that don't work as well are the ones that rise. So I think Quest would be an example of an algorithm which rises very fast. One that rises not as fast would be Max, which has got the red line down here. So based on this, you'd conclude something like, if you were to compare the Quest versus the Max, you say the Max has a lower IDR. Okay, so our goal is actually much more modest than that. We actually just want to say, we have this setup. We just actually want to be able to say, how can we estimate the fraction of reproducible markers? Okay, and when I use the term markers, I'm actually using it like fairly generically. So markers could be genes, markers could be proteins, markers can be metabolites. We'll see that later. But we basically want to have a sense of like, and again, technical replicate means I have the same sample. I've just sent it to 2 labs and have them run the same platform. Okay, and I'm hoping, right, that those 2 things should be fairly concordant. And again, so one of the cool things about this methodology is basically it's going to sort of say, because I'm measuring so many things, I can pull information and say some of the things are concordant, but some of them are not. So I want to get a sense of how many things are concordant. Okay, so here's sort of the setup. So I said markers last slide. I'll call a marker a gene now. So I assume I have n genes. I'm assuming that I'm measuring something. So I have 2 experiments. So it's the same sample. So measured 2 times. And then I'm calling from the first time X, the second time Y. And then I basically convert stuff into ranks within the experiments. So for each gene, you basically get 2 ranks, this one and this one. This is the rank of the gene among the X's, this is the rank of the gene among the Y's. And again, the X's are lab 1, Ys are lab 2. Okay. So here's kind of the intuition. So what I basically plotted are two different pictures here. So this is from lab 1, this is from lab 2, and each dot corresponds to, let's say, a p-value for a gene. So basically what you see is what are black dots and not quite as black dots. We'll call the not quite as black dots gray dots. So you basically see that the gray dots are all sort of clumping near 0, 0. Those are going to be the reproducible genes. The black dots are basically the ones that are not reproducible. Okay? And what we want to be able to say, what we basically want to do is kind of a clustering problem. We want to be able to separate out the black dots from the dots. Okay? So all I've done in going from this picture to this picture is now I've created that rank thing. So obviously this was 1,000 genes I simulated from, and I actually know in this case it's a simulation. I know which ones are the reproducible ones and which ones are not. So of the 1,350 are reproducible. So basically now all I'm doing is basically converting to that rank statistic I told you about. So each point you have the rank from lab 1, rank from lab 2. So what do you guys notice about the picture on the right hand side? What are two things to sort of notice? Yes. Exactly. So if things are ranked near the top, they're highly correlated. And then basically, so this point cloud is quite tight near the bottom left. And as you go along the diagonal, you go from really clumped to much less clumped. Okay. So again, basically, and the way we're sort of formulating the problem is we're basically thinking there's a magic transition point where you go from clumped to not clumped. And what we want to try and do is see, can we come up with a way of estimating the transition point? Okay, so this is basically our statistic. We have that ranked gene, so that for each gene we know it's ranked within X's, within Y's, we're seeing the maximum of it. And we call this, not surprisingly, the maximum rank statistic. And then what we're going to do is basically take that, sort of divide it by the number of genes. So this number is now going to be between 0 and 1. And we're doing something a little bit different than what most people do in statistics. So in statistics, we talk a lot about the cumulative distribution function of the empirical CDF. And that's basically the fraction of observations less than or equal to something. Now, if you look at the formula up there, I've actually flipped the sign around. This actually was called an empirical survival function. And I'll show you hopefully later why this is sort of an easier thing to work with than the empirical CDF here. But basically, we're going to be using that empirical CDF of this statistic divided by the number of genes as kind of our distribution function. Okay, so here's basically the idea. We have that empirical CDF. We call that cat S. I'm actually going to formulate what I'm going to call a theoretical null version of S. And then basically, I'm going to use this optimization problem, basically estimate this pi1. And pi1 is going to be the fraction of genes that are reproducible. Okay. So what you look over here is you basically see this is that empirical thing I defined earlier. And I'm subtracting from it 1 minus lambda, this theoretical S. And I haven't given you the formula for S. It's in the paper. But basically, you can think of it as kind of like a curve matching problem. Okay? So hat S gives me a curve. 1 minus lambda S gives me a curve. And I basically want to see what value of lambda is going to, or an I lambda is going to give me the point where the curves are basically aligned. So how many of you guys work with have used alignment algorithms? This is, yes, I get some hands, which is good. So there's things like, dynamic programming. That's a very classic alignment programming algorithm. This is like way simpler. But really, you'll see on the next slide, basically all you can think of this is basically a curve alignment problem. Okay, in fact, here's the curve alignment problem. So I told you about that objective function on the previous page. So again, this is simulated. This is the simulation from before where we had 1,000 genes, 350 were reproducible. This is looking at that same picture, and this is basically looking to see where exactly is there a drop. And you actually see, so here the minimum here would basically be 0.348, which is a little bit different from, but not too far from 0.35. 0.35 is the truth we simulated here. If you look over here, based on this picture, you have dashed lines and you have this red dotted bunch of dots. The red bunch of dots is that empirical S hat, and the dashed lines are various versions of S. And again, we're trying to get the curves of the S. So basically, what's up here are basically different dashes correspond to different lambda values, and we're basically trying to find the lambda that best matches the dots. Okay, and that lambda value will give you this pi1 estimate. Okay, so that's kind of the algorithm. Now the question is, like, why does this work? So we're going to go do a little bit of a blast to the past. So full disclosure, when I was here in Michigan, the last class I taught while I was here was Bio 6646. Does that class, or 646, does that class still exist? Probably not. The class was on statistical methods for bioinformatics. I had 15 students in the class, 10 were from this department. So anyhow, so this table was from that class. So I had to dust it off, you know. It was on paper or on a transparency or something, and convert it. I'm kidding. It was already a slide. But basically, one thing we talked about in that class was when you have high-dimensional discoveries, like thinking about different error rates. So multiple testing is a big issue. And what I'll talk about now is thinking about how you can convert this MAR idea to think as a multiple testing problem. So basically, this was a multiple testing table that I showed in that class I taught here. So again, you have all these hypotheses you test. And let's say here you have a total of M tests. So you basically do one test at a time. You decide, do I reject or do I not reject? Okay. The number of rejections I make would be Q here. The number of things I don't reject by definition is M. Now, when you think about multiple testing, the decision to reject or not is based on the data. The rho here is basically the truth that we never observe. It's the null. So when I say null, that's short for null hypothesis. Is null hypothesis true or not? Now, if there's statisticians in the audience, they will quibble with my use of null is true versus null is not false. It's been a big controversy, but basically what that means is the stuff inside these capital U, capital V, capital T, capital S, these are not known because we never know the true labels in the row for the rows. Okay. Okay, so based on this table, though, you can define various error metrics. And the one we'll talk about today is something called the marginal false discovery rate, and that's given by this ratio. So basically, Q is the expected number of nulls we reject, and then V is basically of the ones we reject, how many of those is the null true? So you make mistakes with V, you make mistakes in T. So we're looking at V as a function of Q here. And again, for us, if we reject H0, that's the same as calling a gene reproducible. Conversely, if we fail to reject H0, we're basically saying that gene is irreproducible. And then you can think of FDR as roughly the expected number of true irreproducible genes out of the genes that we call reproducible. So again, so if I call them reproducible but they're not, I'm making mistakes, right? And we're sort of estimating a marginal FDR for each gene this way. And so the Q is basically how many genes are reproducible in a certain critical region, and then K hat is a certain critical value, and then V are how many are actually irreproducible, but we call them reproducible. So again, V are the mistakes. So again, the goal of multiple testing is to figure out what are thresholds we need to make sure that we control the FDR. And we're basically doing exactly the same thing here, except with multiple testing p-values, we're instead calling them reproducible genes and not reproducible genes. So that's kind of sort of how we can kind of combine the two problems. So this gets us to our MAR procedure. And basically, here's kind of an algorithm. You get a cutoff hat k based on this optimization problem. And then you basically say n hat is going to be the number of reproducible genes, and the m hat satisfies this rule. And then you basically reject all genes whose maximum ranks are less or equal to n hat. The rule. And again, it's so it's kind of, so it looks complicated. We're basically, so if you're familiar with Q values, you might have heard of the name John Story. He's a statistician at Princeton who sort of came up with that idea as part of his PhD thesis. This is basically applying the Q value algorithm to our problem. So it's basically just, so the trick for us really was figuring out how to, so he uses CDS of P values here. We use the survival function, and then we have to kind of compute stuff here to figure out how to get the FDR. But pretty much it's very much like most multiple tests. So if you've ever used Benjamin E. Hochberg, there's a rule like this for that. If you ever use Q values and compared it to a cutoff, you're using a rule like that. So in a way, it's actually not any different than things many of you have done in your other projects. Okay, so here's the application. So this was a RNA-seq experiment as part of the seq-seq, like one of these quality control projects. So this is obviously going to be with RNA-seq data, and there were 3 platforms, and I think there were 13 labs. I think we won't look at 13 labs, but there were 13 labs in the protocol. So the platforms that were used: Illumina HiSeq 2000, Roche 5.4, and SeqRite Inc. Okay, so what's kind of nice about this study is actually for some of these genes, they actually had done PCR validation. And as you'll see later, we'll actually use that as a benchmark to see, did our method help or not. Okay, so here are a couple labs. And again, the data really come from Qunhua, my former colleague, and her collaboration with people at Penn State. So, so this tells you a little bit about sequencing platforms and from the different labs. So here's 3 of them: Mayo, Penn State, and New York University. So things you notice here. So obviously one thing is the read depth, which is how many reads you get from the machine. That number is like insanely variable. It is super, super high for Mayo. It is super, super low for NYU. Does anybody know what that is? Mayo has better machine. Maybe. I think we're going to try and test the hypothesis here, but actually with the sequencing machines, right? I mean, basically the practical reality is, well, so one thing is the Illumina, the reads themselves, the fragments that come out are actually shorter than 454 fragments. So that's one thing. But the other thing too is, I mean, Mayo might not have better machines, but they might have the PIs willing to pay more to do the sequencing. And that's what's really sort of, I think, being reflected here. Is basically like you can think of, you can actually think of sequencing as having a sort of a cost per read. And basically, maybe what's really being shown here is Mayo's willing to spend more to run the experiment. So I think that those are 2 reasons. Yeah. So anyhow, so here's some other examples. So this sort of told you how they did So I told you it's one sample they sent to these labs. They have the option of doing, do replicates and stuff like that. So you also see here. So again, these are some other universities. I think BGI is probably Beijing Genomics Institute, and WU is Northwestern, and I can't say off the top of my head what SQW is. But again, more data and sort of on the replicates and read depth. And again, the Illumina ones are definitely much deeper reads than the Roche 454 here. Okay. Okay, so here's some results for the procedure. And for the comparison, we actually also looked at comparing it with this copula model that I mentioned earlier. So this was the work of from the Lee et al. Paper from 2011. So we have several comparisons here. So we'll start sort of looking at the let's see. So this is looking at replicates within labs, okay? And so one thing you sort of see here is basically, so let me go through the columns here a little bit slowly. So pi1 is the same for both methods, the fraction of genes that we think are reproducible. Rho hat is basically, of the ones we think they're reproducible, what's the correlation of those? And then finally, this last column, which is FDR less than 0.01 or IDR less than 0.01, is if I were to use a cutoff for estimating the IDR or our new FDR measure and say that, you know, how many genes pass the cutoff. If they pass the cutoff, remember, we say they're reproducible. Okay, good. So anyhow, if you start to look at stuff within labs, one thing you notice is that the fraction of reproducible genes is comparing like these three numbers to those three numbers, it's higher for our MAR method. So we're actually estimating a higher fraction of reproducible genes than the copula approach. If you look at the next column, though, you get a sort of a paradoxical thing, which is, again, it's like, you know, it's in the third decimal place usually, but like the correlation from the copula method of the reproducible genes is slightly, almost the same, but maybe in one case slightly higher. And then if you look at how many genes, again, because we're calling a lot more genes reproducible, we're also calling a lot more, with the rule, our rule tends to call more genes reproducible than their rule. Okay, so just a couple reminders. The other thing to notice here is that the pi1, again, this is the same sample, right? That's been run at all these different labs. And you basically see the pi1 changes a lot. Because you go from like 0.566 for NYU up to 0.851 for Mayo. So one thing we sort of concluded from this experiment, and we did some other analyses we'll talk about shortly, is that the read depth affects the reproducibility a lot. And this probably makes sense, right? The deeper your reads, the deeper your sequencing is, the more reads you get, the more reliable your findings are going to be, right? But like I said, it comes with a catch. The more reads you do, the more money you have to spend, right? So it sort of gets to a sort of a classic kind of thing and that we think about a lot is basically what's going to be the bang for the buck in terms of benefit. And again, just to remind you, like this was the one, if you go back a couple slides, so 600,000 roughly for the NYU and then 150 million for the, so 600,000 gets you a pi1 of 0.566 and 150 million gets you a pi1 of 0.85. So again, so that's sort of one interesting finding from this analysis. The next one is now we're going to compare the same platform at two different labs. So that's 4, 5, and 6. So if you do that and look at Mayo versus Beijing Genomics Institute, you sort of see super high concordance with Illumina. And again, with PSU and NYU, you get slightly lower, and then the lowest is the Roche. Now, what I found interesting about this analysis is that we're actually getting something very kind of non-intuitive, which is that the between lab reproducibility is actually slightly higher than the within lab reproducibility. So anyone have thoughts as to why this might be? But unfortunately, this is a little story. This paper got published, and we never actually addressed it in the paper. So we were aware of it and had no great explanation. So yeah, just something to know. This is not what you should be expecting. You should be hoping that with the within lab reproducibility would be higher than the than the between lab. Okay, so this is the validation. Yes. Good question. So I think yeah. So Qunhua has a paper where she did her approach, and they did a similar kind of study with I think it was with either ChIP or ChIP-seq, same sample, multi-lab thing. I think they got very similar. I think they got similar kinds of things. So I mean, I only have my N of 2 here. So the 2 times we've seen this phenomenon. Okay. So this is the validation I told you about. So I told you for some of these genes we had gold standard measurements for PCR. So what we then basically did was we had different comparisons, and we basically looked to see, like, of the comparisons of the things that were reproducible, how many of those were PCR validated and what were their ranks. Okay. And again, with ranks, so here what you want is, like, the lower your average rank, which is going to be these vertical lines, the better your method is doing. So we have 6 comparisons from the previous page, and we're basically looking at so the one on the left is our non-parametric approach, MAR. The one on the right is the copula, or the one by Lee et al. 2011, and you basically see there's some, for this one is close, this one is close, this one gets bigger, and number 4. The copula method didn't find any of the PCR genes as being reproducible, and same with 5. So out of the 6 comparisons we did, only in the 6th comparison did the copula method do better. But the other thing you start noticing is that, again, we were calling more things reproducible. Again, just because we're calling more reproducible, they were also more likely to be sort of PCR validated as well. So the number of points we seem to be doing better, and then the average of the rank, in most cases, we're doing better. And again, this is always the challenge of bioinformatics, right? We can make predictions all the time. The question then becomes, how do you validate the prediction to be able to make the claim that your algorithm is doing better than other ones? So, the questions about any of this so far? Oh, good question. So we computed this as kind of, so again, all the algorithm really can do is do this part. Once you get that right, you then make a decision, what's reproducible, what's not. And then you can compute summary statistics. So, I mean, this is very much tied to this. And so I don't think, because they're tied, I don't think you can say one's, like, more important than the other, because they kind of, like, the first one depends on the other, right? I mean, basically, sorry about the Shiny app issues here, guys. So let me put it this way: I think by chance, the fewer genes you call reproducible, the higher your row will be. Because again, go back to the picture. The row is something about the row has something to do with this picture here, right? And what's the correlation? And again, sort of like if I take if I look at small subsets near the origin, I'm going to have artificially high correlation. If I make my ovals bigger, right? At this point, if I start to put in more and more black dots, my correlation goes down. So the correlation is going to be very much a function of what I call the cutoff and what I call reproducible and not. The cutoff depends on pi1. So they're sort of linked. I'm not sure you can decouple the 2 parameters the way you're thinking about it. Other questions? Yeah, good question. So this is sort of so obviously this is one example. The basic fundamental question we're asking is, like, how correlated are 2 things, right? So, and looking at correlation setups like this, where you are measuring on a sample a lot of things, right? So, this is an example with genes. Other things I work on things like brain imaging. So brain imaging, you have voxels, and you have measurements in voxels. And then you might want to know, like, if I've measured the same brain twice, 2 times, how correlated are the voxels? It'd be the same setup. So it's very much, so what this will get at, right, is something about your instrument that you're using and how reliable is it in the measurements. And that's a very general phenomenon, right? You guys work with all kinds of machines. You work with mass spectrometers, you work with sequence experiments, you work with Fitbits, you work with monitors. You want to know, this is getting something at basically what's the measurement error of the machine. And this will sort of start to tell you that. Good question. Both, right? I mean, they're sort of, again, yeah, they're those, it's all a protocol. And yeah, so for this one, for sure, how they handle the sample would also affect things. Good. Other questions? Yeah. 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 Right. Great question. So, yeah, so for this kind, I mean, again, this is a pretty special setup, and I'll get to the example later, but I think in most situations, unfortunately, we don't have technical replicates, right? I mean, I'm sure most of you guys work in studies where you're looking, especially right at a medical campus, where you basically have people with and without disease. You might be only able to get one measurement from each of those people, right? So, so potentially, yeah, it's a good question. I mean, I think, so obviously, I'll make my stats pitch, which is, having this kind of a experiment done as part of your study can help you get a sense of what your this measurement error variance I was talking about is. And potentially, so if I see a pattern like this for the same sample, and then let's say I get 2 different samples and look at this, and I do the same kind of thing, I can use this to calibrate what I should expect to be null in the 2 different sample case. And then deviations from that would basically be real signal. So that's kind of how I would eventually use it. But again, the other things we can do as well, right? In a lot of situations, we don't have this kind of reference sample thing. And I'm sure you guys probably do lots of things where you create pseudo reference samples by looking at medians of measurements across all your samples or something. So, and again, that's a very standard thing people do with bioinformatics experiments. So. Okay, so I have a couple more examples. This is more the metabolomic stuff. So Al and I, we've been working as we're both PIs on separate UO1s as part of this Metabolomics Common Fund Consortium. So basically, I sort of pitched this methodology to my group, and they were like, we were talking about what kind of examples could you do. So we actually have a few examples we tried this on metabolomics. I don't have much slides about the data issues, but I'll tell you one catch. What I showed you earlier was basically the cleanest thing possible. So let me tell you before I will show you the findings, I'll just sort of tell you some of the data issues we've been sort of dealing with. So with metabolomics, you run the same sample twice. You might not see the metabolite both times, right? So you have a missing mechanism. Other things, and again, there's lots of reasons for that. Metabolomics, like any instrument, has a detection limit. So especially for low expressing metabolites, you can imagine that the value might fall below the detection limit. You also might get, when you run the experiment, you might aligning, and with metabolites, these are going to be mostly targeted, annotated metabolite experiments, untargeted but annotated. So what you basically get for each from a matrix is basically going to be a mass charge ratio, retention time, and for sample abundance value. And again, there's always an issue, right? That the same sample run twice might give you different mass charge and retention time values. So how you deal with that's another issue. So we have issues like that we're sort of grappling with, which actually makes using this MAR methodology way harder on metabolomics. So this is much more work in progress, but I just want to show you some results to show you that we've tried it on some metabolomics data. So the first one is kind of like what we had earlier with the RNA-seq experiment. So we had one sample, and then what they did with that sample is they basically send it to, so this is operator 1, operator 2, operator 3. Each operator ran it, or operator 4, and each operator ran it 3 times. So that's one example, and that's from this core at Colorado that Nicole Reisdorf directs. This one is a little bit more interesting. We actually have a lot of subjects, 131 subjects, and again, they have different, some of the, I forget what disease we're looking at, but okay, good. Good. So this is, so subjects they come from different populations, but for each subject, we've measured them 3 times. So it's a mix of biological and technical replicates. And this last one is only, same kind of study as well. So, so basically we tried it. So here we did basically no processing. And what you basically find, it's hard to tell. So basically the scale is from 0 to 100. And we're basically looking at what are percent reproducible metabolites for the batches. And again, remember, it's the same sample. So it should not be changing a whole lot. So there should be pretty high reproducibility. And you basically see if we don't do anything to the data, we're getting reproducible values, these pi1s, we're getting around 20 to 35%. If you remember back on the CQC experiment stuff, we were getting 0.6 to 0.8 or 0.9. So we're actually much lower here. And then we did it for the spike-ins. So this is at the top level, basically, of the hierarchy with all the samples. This is doing it for the spike-ins before you, yeah. So that's the middle layer, and then these are for all 27 replicates. And what I should say here, so if you remember, the statistic took 2 numbers and took their max, right? But if I look at the design of the experiment, at this level, I have like 27 numbers. Up here, I have 3 numbers. So one thing we basically had to do was come up with summaries. So what I'm showing you on the next page are basically pairwise comparisons. So like for the 27 down here, I'm going to have 27 choose 2 pi1s. Up here, I had 9. I want to have 9 choose 2 pi1s. So that's yeah, that's sort of what's so again, what you basically see though is in general, at the higher layer, not very good, still not very good at the middle layer, and then starts to slightly get better at the bottom. Still not good. So basically, one of the sort of conclusions is basically you have to do something. You have to do imputation and filtering if you want to make this sort of be more reliable for metabolomics. That was not something we had to worry about with RNA-seq. Okay, this is the other one. So datasets 2 and 3. So just interestingly, this one you start to see better performance, and in this one we have now done some filtering and imputation. So what was between around 20 to 40 before, you're starting to see now more values more near 60 to 70 for the most part. And then down here, now I've mixed, so this is technical samples. So these are basically for the 131 subjects looking at everyone's technical replicates. So you look at stuff within the subject. So now I'm picking 2 samples from different subjects. And what was 60 to 70% for the technical now becomes, closer to 40, but now potentially I'm mixing samples from different people. So again, you can think of like, sort of using this to calibrate to this and then figure out what, the differences might be actually due to biological difference. And then this last one, unfortunately has no technical really, but just biological. This is one with 1,100 subjects. And you basically see here, you're getting like really, really, really reproducible metabolites. Now, what I can tell you about dataset 3 is basically this is one that we got from Metabolon. Those of you metabolomics know that Metabolon is probably one of the major industrial companies in this space. And basically this dataset we got pretty much processed by them. So it was filled in, no missing values, everything annotated. So I forget how many metabolites there are on this one, but the catch with this one basically is we never got the actual sort of raw data. We have no sense of what steps they took to get to this. But again, it looks quite quite high here. So yes, that's a good question. I've asked this to people that actually run the machines, and they seem to give me the suggestion more that it is variable. There are, I mean, with these machines, right? You have drift effects and things like that, which would sort of impact the detection limit. That's a good question. Let's see. In this one, we did not throw them away, and they were zeros in the data set. Or we call them absent. Correct. So again, the work we're looking at is basically and this is work in progress. We're basically trying to figure out, like, what if we start to try to model the zeros, and if we do that, can we? And again, sort of, I didn't show that today, but basically, some preliminary stuff suggests we'll do a little bit better. But again, as you're well aware, Brenda, like, this basically gets into multiple mutation kind of ideas. So we're thinking about, if we want to do a multiple mutation here, and think about how to estimate things after multiple imputation, how do you think about combining analyses, estimating imputation variance? So you could do left sensor data analysis. That's the other approach. So we have a couple people that I didn't mention today, but there's a postdoc looking at using left sensor approach to this as well. Good. So this is the other question. This was the other part. So this is yet more things. More sort of analyses here. So what I showed you before were metabolites across subjects. This is asking the question differently, where I basically transpose a subject and a metabolite, and to say, like, how many samples are reproducible for a given metabolite. So this is getting at the question is, how reproducible are individual metabolites? And again, we can do that here because we're basically flipping the roles around of what's the metabolite, what's the sample. So when you do that, you basically start to see here that for the technical replicates, you have this very interesting pattern where basically some metabolites are super reproducible across everybody, and then some just aren't at all. And again, this sort of fits into that MAR idea that we've been talking about, right? That there's going to be a fraction that are really reproducible and a fraction that are not. That is for so dataset. This is for technical replicates. Here you see a lot less concordance. But again, what biological replicates means are samples are coming from different populations. So again, you could use the idea of calibrating this distribution to that distribution to figure out what might be an important metabolite to follow up. And then this is for the really big data set. And again, you sort of see, this is just biological replicates. So you see, again, not the greatest correlation. Okay. So to wrap up, really this goes back to a sort of in biostatistics, just a very classical topic of assessing agreement. And there's things like Cohen's kappa and rows and things like that to do that. And I basically say this is a fairly flexible way of doing that, and it's sort of taking advantage of the fact that you have a lot of measurements on samples. Again, when I went to grad school, I grew up in the era of, like, you measured, blood pressure, height, weight, kind of stuff on people, and now you measure stuff, SNPs on 1 million markers, or RNA-seq on thousands of genes. So sort of using the idea that you have high dimensional data, you can sort of model agreement this way, and sort of here's some future directions we're looking at. So we sort of want to start to look at simulations for how well the procedure works. And again, sort of one thing that's always kind of bugged me is the statistic is a maximum of 2 numbers. In most experiments, you have way more than 2 numbers. It turns out like that I mentioned an S hat and an S earlier. It's really easy to find S when you have 2 numbers. It's much harder to find the S when you have a lot of numbers. So we've been talking a little bit about how we extend the Mars to handle more than pairs of subjects. So to wrap up, I just want to acknowledge funding. And again, this took place at 2 institutions over a lot of years. So there's a lot of funding involved to get the work done. And I'll just conclude with just a paper sort of describing the methodology. So thank you.
Thank you very much for this great talk. Are you going to implement the calibration? You are talking about the calibration?
Yeah, that's work in progress. I mean, yeah. Right now, we're sort of finishing up for the metabolomics, just the issues that we talked about, like if you want to do the imputation, how does it affect things? And then the next step would be looking at it'd basically be a differential expression analysis. So that's the next step, yeah.
Okay. Other questions? So actually, when you complete this sort of, work and develop it, is that going to be part of the tools that we are developing for metabolomics? Because I think it's, as you have shown from your examples, I mean, for metabolomics, it's much bigger problem than for
exactly,
other fields. Basically.
That's the hope. I mean, again, so just to sort of say a little bit, when Al and I put in our proposals for this RFA, one thing we had to sort of specify was kind of the software plan. And again, unfortunately, this wasn't the best example today, but this is what I'm actually showing you is a talk using the Shiny R Markdown command in our. Part of what I want to do is to make things a little bit more reproducible presentation-wise. Unfortunately, it was also stopping-wise, which is So the plan basically is when we do these things, we basically have a multi-level system of how we think about code release. So for sure, we'll have at a minimum we'll have sort of our packages up on GitHub at a minimum. And then we also have, as part of the grant, we actually hired a computer science undergraduate major who's working on helping us sort of like do some software development. So he's ultimately he's also going to start to build things like Shiny apps based on this stuff. Yeah.
Question.